If you see them in a bunch, that's low fall. If you see them all over the surface, that's peritrichous. These are all in the notes. I'm just summarizing them here. If you see them one on each side, I don't know how to explain, but they're amphitrichous. Now, so this is what they look like. The basal body is rings on a rod. You have the L, P, S, M, C. These are the rings, right? This is, you have the hook. I'm throwing on top of them each other. And then you have the filament which extends all the way out. I mean, hopefully you're following. All of these rings are found in gram-positive or gram-negative. All of them are found in gram-negative. Which ones are only found in gram-positive? The bottom ones. So gram-negative has all of them. Gram-positive only has the bottom two, SM and C. So SM and C are only in gram-positive. Sorry, I should take that back. SM and C are the rings that gram-positive have. Gram-negative have all of them. Okay? So clearly, which ones are going to be essential for motility? The bottom ones, because the top ones are lacking. Don't worry about what they're called. L is, don't worry about it. Hook is boring. The filament is the long one, which is made up of what? Flagellin, a protein called flagellin. That's pretty much it. Now you do have to know how they move. Remember, they rotate, right? And when they rotate counterclockwise, the bacterium is running. If they rotate clockwise, the bacterium is tumbling. That's chemotaxis. So clockwise is a tumble, counterclockwise is a run. Run means it goes long, straight line. Tumble, it jiggles about and then starts another run, on and off. Okay. That's pretty much it for flagellum. And then please remember the types of motility, right? So for example, what I described as running and running and tumbling, that's swimming, literally swimming. So it needs an aqueous medium. But they can also glide on a surface, right? Which means they secrete slime and literally slide or glide on a surface, and they can swarm on a surface. So they can swarm, they can glide, they can swim, or they can just remain without movement. The last big topic there is endospores. So here, again, remember the names of the organism, right? It was the endospores. And the endospores are almost exclusively in gram-positive, right? So what are Bacillus, Clostridium? These are the two big ones. And then please remember the diseases associated with them, because we've discussed them throughout. Bacillus, we've only discussed one disease, which is anthrax, right? Anthrax, Clostridium, there's a bunch of them. Clostridium has botulinum, tetanus, gas gangrene, that was Perfringes, this chapter, the last chapter. Okay, so what makes endospore? There's one chemical we discussed in endospores that makes them very heat resistant, and that was, remember? Someone stops you on the way out and asks you which chemical?
Calcium.
Calcium dipicolinate. That could be 1 point right there. So it's in the notes. Don't. Please don't forget it again. Excuse me. So, yeah, what makes them heat resistant? Calcium dipicolinate helps. They have very little water. They have a lot of coats. And please remember, if the bacterium is making a spore, it's called the sporangium. Okay? What's the outermost coat called? Right? When you have an outermost coat, outer means exo, so it's the exosporium. The notes are there. There's two other forms of spores we discussed. There's one called exospores, which are produced by Streptomyces, and the cyst, which is produced by, well, many, I don't know which one, I think I gave Azotobacter. And then one more concept could be true and false. As we discussed them in class, bacterial endospores are not reproductive, true or false? They're not reproductive. True, because a spore will give you the vegetative cell, which will give you the spore. There's no increase in number. There's no... Okay, questions about 3. 27 is the antibiotics. Now it is a relatively small chapter, so it's not... It probably will have about, I would say, maybe 9 or 10 questions. But they are going to be straightforward questions. As was the case on exam 2, you don't have, for cell wall, first of all, there might be 1 or 2 questions from the introduction right there. What are the organisms that produce bacteria? What are the organisms that produce antibiotics? We discussed 3 big ones. The top level would be actinomycetes, including streptomyces. Then you have the molds, including penicillium. And then you have bacteria other than actinomycetes. And the main one is bacillus. Right? So that could be a rank right there. Which one is first, which one is second, which one is third. What is an example of a narrow spectrum? We discussed isoniazid. Isoniazid is exclusively, well not exclusively, almost exclusively towards tuberculosis. Right? Tetracycline, on the other hand, is very broad spectrum. Almost anything is targeted by it. From there on, you would have to know the antibiotics based on groups as to what they target. Right? So for example, with cell wall inhibitors, don't worry about which step. Don't worry about that, how bacteria make their cell wall. But please know which are cell wall inhibitors. You are going to get questions like this. So for example, this is a question here. All of the following are cell wall inhibitors except, so I meant list 4, it's for the sake of speed, I guess. Okay, so all of the following are cell wall inhibitors except penicillin, bacitracin, streptomycin, cycloserine. And the answer, if you don't know it now, hopefully by Tuesday morning, it's streptomycin. This is cell wall inhibitor. That's a beta-lactam. Not beta-lactam, but cell wall inhibitor. Beta-lactam. Streptomycin inhibits cell, not the membrane, the cell's protein synthesis. It says protein synthesis inhibitor. Okay? So please know which target the cell wall, which target the cell membrane, which target protein synthesis. With protein synthesis, don't break them down between 50 and 30. Please know, is it cell wall, is it protein synthesis or not? Right? So for example, there's a bunch of them: tetracycline, streptomycin, chloramphenicol, oxazolidine. There's a bunch of them. They're there. Nucleic acids, you would have to separate them between DNA and RNA. So please remember which one is a DNA inhibitor, which is an RNA inhibitor. We talked about rifampin being RNA. Cell membrane polymyxin. You can make flowcharts, you can make note cards, whatever. You're going to get 8 or 9 questions about antibiotics. And they're going to come in this form. All of these are except, or some would be more straightforward. Which of the following inhibits the cell wall, or breaks down the cell membrane, or whatever. There are also some that we discussed as anti-metabolites. And here we discussed 2. We talked about sulfur, actually more than 2. But sulfur drugs inhibit, there are 2 enzymes that I discussed. Sulfur drugs inhibit the dihydropteroate synthetase, and trimethoprim inhibits the dihydrofolate reductase. So know these 2 enzymes. That could be 1 point right there. Dabzone. Does Dabzone ring a bell? Dabzone is no, we're going to get to. Dabzone is anti-leprotic, which is anti-mycobacterium leprosy. That's also an anti-metabolite. So again, isoniazid, tuberculosis, Dabzone, leprosy. With fungals, same thing. Now those 9 or 10 questions that I mentioned are going to include some antifungals. With antifungals, do the same thing, right? There's only, I think, 6